Good. So my name is Lennart Voortman, and I will give an overview of the research facilities and primarily focusing on the preclinical sciences. So I'm the head of the light microscopy facility, but what I'm presenting here is more based on what's happening in the research facility assembly. So the research facility assembly is something we started about a bit more than a year ago. With a couple of research facilities, which slowly grew into a bit larger, what we now call the research facility assembly. I should emphasize this is really a bottom-up initiative where our main goal is to learn from each other in terms of organizational as well as practical matters. And as Biobanking also said, it's really important for us to improve our visibility both to our users, but also within the organization. We're trying to do this by just making sure we coordinate things better, at least amongst each other. So at the moment, this is like a 6 weekly meeting. So it's hard to get an overview of how many facilities there are within the LUMC, but now we have at least 15 facilities joining, but also the fundamentals for innovation and the directorate research to align everything together. So if you want to know more or you want to join, everything is really open, but please drop me an email or drop my office. Always really happy to welcome new people. So I wanted to start by saying that this presentation, even though I'm presenting this, really is a collaborative effort from all the people from all the different facilities. So it's really a lot of different facilities contributing pieces to this presentation. So to give a bit of an outline of what I'm going to present, it's a bit challenging, I would say, because on the one hand, we have a lot of different research themes, and somehow we're trying to connect this with all the different research facilities. So I try to make a bit of an outline, but it's going to be a bit of what I say is a bit of a bumpy ride between the themes and the facilities. So bear with me, but I hope in the end you'll have a bit of a better overview of what the different facilities can offer and how this connects to the different research themes. Okay, so we start this journey from the research theme of immunology. And here I'm showing you on the left. I'm just going to put my pointer to laser pointer. A recent publication studying the immune response to coronavirus, and then specifically looking at what's happening in the upper respiratory tract, so the nose. And this study was like a very large collaboration involving many different authors from all the corners of the LUMC. But just as important as all these brilliant scientists, also the support of the different facilities that supports large studies like this. So on this slide, I'm just highlighting 3 different facilities: the flow cytometry core facility, the biosafety level 3 hotel labs, and they're of course essential to this type of virus studies. And also the biobank, which stores the material of the different beat COVID studies. And this is also essential to also enable future research questions to be answered with this type of material. Okay, and before we have a bit of a closer look at different research facilities themselves, I want to highlight one thing, and which is more or less how the facilities and the research groups also feed back into each other into like a circle of innovation. An example is this Cytoexplore, which was actually developed in the research group of Biobanking, but is now also, again, offered as a part of the work done by the flow cytometry facility to analyze the data. So here, the facilities and the research groups really strengthen each other. Okay, so let's have a bit closer look at the flow cytometry facility. The flow cytometry core facility provides flow cytometry, mass cytometry, and cell sorting to a wide range of users. But also education as service is a key to what the facility does. These services are offered, of course, to a wide variety of users from the LUMC, but also quite a lot of external academic groups, and also a number of companies from the Leiden Bioscience Park. In terms of equipment, the FCF houses all the advanced flow and mass cytometry instruments of the LUMC. And this lineup currently contains over 24 different instruments for both research, but also diagnostics. Then on the slide before, I also mentioned the biobanking. And this is an essential infrastructure and support to collect, store, or distribute biomaterials for future research studies. So throughout the years, this biobank, different biobank collections from the LUMC have contributed to essential material to many important studies. So here, of course, the famous Leiden factor V discovery, but also worth mentioning the already 25-year-long effort on the arthritis cohort, which includes material more than 4,000 patients, and this resulted in many different studies. I hear about 170 different publications, and this is just 4 examples, and also the work of more than 20 different PhD students supported by this type of material. Then I would like to jump back again to one of the research themes, and this time it's the cell, tissue, and organ. And I think this is a really cool study that brings together many different research areas. This is a study from the nephrology department, in which human pluripotent stem cell-derived kidney organoids were transplanted in mice under a renal capsule. Then by imaging these organoids after transplantation through this window, they confirmed that over time, the organoid was actually vascularized by the host, so the mice itself, and actually showed functional glomerular perfusion. But you also see in a study like this that a lot of different facilities support research questions like this. So now I wanted to jump into a bit more details about IPS Hotel. Then the LUMC IPS Hotel offers generation of research-grade IPS cells from different tissue sources using state-of-the-art reprogramming techniques. And the services offered by the IPS Hotel is, of course, training and support, but also, of course, gene editing to generate isogenic controls or knockouts or reporter cell lines. And essential, of course, the differentiation of these IPS cells into different cell types, for example, endothelial cells, cardiomyocytes, and macrophages, etc. Also important for a centralized facility like this is the quality control. And quality control is in turn also supported by work done by a different facility. And this is now the LGTC. So the LGTC, the Leiden Genome Technology Center, is the genomics and transcriptomics facility of the LUMC. The LGTC is specialized in single cell sequencing, but also long read sequencing, and also latest and greatest equipment from a couple of different companies. 10x Genomics is Dolomite Bio, PacBio, Nanopore. But the LGTC also offers services, including Sanger sequencing, DNA, RNA, quality control. But they're also always available for general questions about DNA, RNA sequencing. But they also go much further in really supporting researchers with custom sequencing protocols, really involved with the development and implementation. Then a bit of a different facility is the PDC Mouse Clinic. It's a really, I say, a key enabling facility, which really, together with the associated transgenesis facility and the preclinical imaging facility, really enables animal models to be studied in the translational research. So I think they're essential to the well-regulated animal studies at LUMC. And also by centralizing these within facilities, It also enables responsible animal experience, of course, along the guidelines of the replacement, reduction, and refinement. So the facility offers a wide range of services, of course, from the creation or the breeding of the mice. They, of course, take very good care of the different animals. But they also take good care of all the scientists. So they really support the scientists with the tools needed for these animal studies. This, of course, leads to many papers being published together with the PDC. Then one of the tools I want to also have a close look at the preclinical imaging, and this is really a tool used in these animal studies, and the preclinical imaging facility offers a range of imaging modalities for structural, functional, or molecular imaging of living animals. The techniques includes MRI, CT, SPECT, optical, but also ultrasound. What you see here on this slide is an example of a study into cerebral amyloid angiopathy, so CAA. And I would say there's really like a mix between the neuroscience and the cardiovascular research. There's also two different research themes. It also pieces together like results from animal models, but also biobanking material. What we see here is results from ranging all the way from MRI to even electron microscopy. So also here, different facilities collaborating together to really provide the services needed for and support the research questions. So then we're continuing in this research theme of cardiovascular, and here I've highlighted a research paper which received quite some attention in the media, also on the LUMC website, of course. And in this paper, the researchers from the LUMC showed that for the first time how to immortalize cell lines of human atrial myocytes. It's really, really nice and what do you say is really useful innovation, and also again supported by the facilities available here at LUMC. And now let's have a look at the light and electron microscopy facility. And the light and electron microscopy facility actually consists like of two parts, one part light microscopy, one part electron microscopy. And later in this seminar, Roman Koning will tell you everything about the electron microscopy part. So here I'm just showing a quick overview of what the light microscopy facility does. So to me, the main thing what we do is support many different users from all the LUMC. So last year we had 375 unique users, and together those users made a reservations for more than 40,000 hours of microscope time. And to get all these people using the microscopes, for every microscope, every person also gets a training session. So we're also supporting this with a lot of different training sessions. Also, one thing I would like to mention is because I think it's really exciting. We just installed a new microscope, which is called a light sheet microscope. And this is mainly if you're interested in automated imaging of large three-dimensional samples, which normally don't really fit in a light microscope, then please get in touch. It's really, really cool new innovation. Okay, then another facility which I wanted to show is the CPM, so the Center for Proteomics and Metabolomics. It's I would say a large research facility, and collaborates also with many different research groups throughout the LUMC and also outside of the LUMC. At CPM, they really have the latest and greatest equipment, but also very qualified people and researchers on metabolomics, proteomics, glycomics, and mass spectrometry imaging, mass spec imaging. Some of the key equipment here is the chromatography, the mass spectrometry, and the NMR spectroscopy. And due to this, these are quite involved techniques. So the CPM also has a really strong research component and builds also close collaborations with researchers from different fields inside of the LUMC, but also outside of the LUMC. Now we're jumping to another facility, which is the protein facility. And the protein facility started a couple of years ago. And they support many different types of research by the generation of very specific proteins. And here I'm showing two different examples. So on the left side, we have an example of what we call early drug discovery work, which is a new inhibitor of DNA replication in a pathogenic bacteria. So here it's really also combined with structural studies using cryo-electron microscopy to really reveal this novel mechanism, which more or less stops this DNA replication machinery by really blocking it with a new inhibitor. On the right side, an example of a bit later stage drug research. Also structural characterization of a novel antibiotic, this time against tuberculosis. And this is now more or less ready to go in the phase 1 clinical studies. And here you also see the collaboration between the Leiden Bioscience Park and the different facilities. Yeah. Then, related to the protein facility is the peptide facility. Because the peptide facility produces custom-made peptides or small proteins, and they do this in using a really chemical methods. And because they use these chemical methods, it also allows them to really design these into very specific or modify them, and this in turn allows them to have these peptides turned into research tools. And here we have two specific papers that use these tools to really profile the activity and the preference of the SARS coronavirus protease. It's really exciting what they can do by really controlling the chemistry of creating these peptides. Then another facility is the viral vector facility. It's a relatively small but very focused facility which really supports efficient gene transfer. So as you can see on the slide, they have a huge library of lentiviral short hairpin RNAs targeting both mice and men. Also a large library of lentiviral CRISPRs for human genes, as well as plasmid-based cDNAs. And by doing this work, over the years, they've supported more than 15,000 batches. And this example about more than 200 papers is just on the short hairpin RNAs alone. So this is really a facility which provides very targeted support for these lentiviral vectors. Then together with all these facilities and all the research within LUMC, we all generate, I would say, a lot of data. We generate a lot of images, sequences, everything generates data. So this is where the data analytics facility really becomes important. Because the data analytics combines knowledge and expertise on the really state-of-the-art methods for data analysis. And this facility is really like a collaboration of experts from different departments and research groups. As you can see, you're also in there's different puzzle pieces within this puzzle piece. And what they do is they organize workshops on different subjects. So, for example, prediction models, artificial intelligence, of course, and also causal inference. So every year there is also a call for small-scale research projects that involve novel methods for data analysis. And this way it really forms collaborations within the LUMC with one of the data analytic experts. So last year, 5 projects were granted, and they're now performing research on different topics. By really doing this in a joint collaboration, it also leads to many publications, also on a very large range of methods for data analysis. And here is just a couple examples on the slide. So the last research facility I would like to discuss is the Center for Cell and Gene Therapy. The services provided by this facility are like full translational support program and manufacturing facilities for advanced innovative therapies, both in preclinical and clinical phase. One of the main product groups which is developed and manufactured by the facility are the cell and gene therapies products. Examples for this are, for example, the T-cell products for the treatment of viral reactivation of malignancies. Also gene therapy products for the treatment of leukemia, as well as stem cell products like beta cells for the treatment of diabetes or a GMP-grade hIPSC cell line. Well, next to these cell and gene therapies, the CCG also supports generation of challenge agents for controlled human infection models. One example is a full release process for schistosomyiasis, but also the manufacturing of GMP-grade coronavirus, which can be used in a controlled human infection model. Well, finally, the CCG also provides regulatory support. It's really necessary for the translation of these products to the clinic or for use in clinical studies or also the regular patient care. So this was, I think, a bit of a bumpy ride through all the themes, facilities, a lot of different facilities. So if you want to find out more, please also have a look on the LUMC websites, on the research facilities. I realize this is kind of outdated and not the best website at the moment, but we're also working on that part. So we're really working also on the website and really improving the visibility of the facilities.